Hey there, all you cool cats and kittens. Welcome back to Bio 150 Lecture. So, we're going to pick up right where we left off. We stopped talking about evolution and this process of microevolution that can eventually lead to speciation, giving rise to all the biodiversity that you guys have learned about. And so now we're going to pick up talking about the major taxonomic groups that provide and compromise Earth's biodiversity, how these groups are related to one another, and the traits that define these taxonomic groups, and probably most importantly, why we should care about these different taxonomic groups. So there's three major domains that compromise Earth's biodiversity. And remember, when we talk about a domain, what we're talking about up in the right in red there is the group above a kingdom. So these are the largest sort of taxonomic groups. And those are the Eukarya, the Archaea, and the Bacteria. And the Archaea and Bacteria together constitute the Prokarya, or the prokaryotes. And when we look at the phylogenetic relationships of these three major domains, we can see that the Archaea and the Eukarya are more closely related to each other than they are to bacteria. Yet we group the Archaea and the Bacteria together into this group, the Prokaryotes. So your first question is, true or false, the prokaryotes are a paraphyletic group? If you said true, that they are a paraphyletic group, you were correct. So remember, a paraphyletic group are two or more taxa, their common ancestor, and everything down the lineage from that common ancestor. So Eukarya and Archaea would be an example of a monophyletic group. If we snipped it right here, this would be a monophyletic group. Whereas a paraphyletic group is a group that doesn't pass the one snip test. So for example, if we snip the tree right here, we could get a monophyletic group, Eukarya and Archaea. But to get just the Archaea and just the Bacteria to fall off, you'd have to snip the tree here and here, or you'd have to snip it here and here. So you'd have to snip twice to get just the Archaea and Bacteria alone. Thus, they are considered to be a paraphyletic group. Another question harkening back to this idea of taxonomic groups, just as a review. Which of the following does not list taxonomic groups from larger to smaller? Note they don't have to be in exact order, just from larger to smaller. So like, is domain a larger group than a phylum, and is phylum a larger group than a genus? So go ahead and answer that question. Once again, this is just a review. If you said 2, you were correct. So classes are larger than orders, and so this was the incorrect grouping. So that was just a quick review of the different taxonomic groups in their hierarchy. So Prokarya, as we said before, is a paraphyletic group. It doesn't pass the one snip test. It is not a monophyletic group. So if this group doesn't reflect the evolutionary relationships of these two domains, Archaea and Bacteria, why do we group them together? So understanding that prokaryotes is a descriptive term, not a taxonomic group, not a monophyletic group that describes their evolutionary relationships to each other. The reason we put them together is because they share a suite of characteristics that make them prokaryotic, that is, distinct from the eukaryotic group. So let's talk about what those characters are. So when we think about the Archaea and the Bacteria in general, typically you're thinking of very microscopic organisms. So the pictures on the right just show you some examples of what Archaea and Bacteria look like. So for example, for bacteria, you've got little microscopic things like E. coli and Streptomyces. For the Archaea, you've got this thing called Sophalobolus and Methanocerina rumen. And in these two cases, basically you've got microscopic organisms. So that's what you should be thinking of, or you should have in your mind's eye when you think of Archaea and Bacteria, as opposed to the eukaryotes, which are typically what we think of as large multicellular organisms. So things like fungi, things like animals, things like plants. These are all examples of eukaryotes. But it's important to also note that eukaryotes also have some single-celled groups. So things like protists and diatoms that are microscopic single-celled organisms that are still considered to be eukaryotes. So these, for example, are some beautiful diatoms, and these are once again an example of eukaryotes. And if you looked at these things on a picture to the naive eye, you might say these look like bacteria or some sort of prokaryotic organism to me. But these are in fact eukaryotes. So what are the traits that define eukarya versus prokarya? What are the traits that separate these two major groups? So if you look at the cell structure of prokaryotic versus eukaryotic organisms, you get a lot of clues to what defines these two groups. So on the left side here, we're looking at eukaryotes. Eukaryotes are called the true kernel cells. So eu means true and kary means kernel, as opposed to the prokaryotes. So pro means before, and kary once again means kernel. So this roughly translates to the true kernels versus the before kernels. And if we look at eukaryotic versus prokaryotic cells, a couple of trends come up. Typically, eukaryotic cells are much, much larger. So this is an individual eukaryotic cell or a cell of a eukaryotic organism. And as you can see, it's much bigger than this prokaryotic cell. And really, this should be even smaller than this. Another important trait of eukaryotic cells is that they have membrane-bound organelles. So things like a nucleus, things like mitochondria, things like ribosomes, things like vacuoles. These are all examples of membrane-bound organelles, and that's something that's unique to the eukaryotes. In addition, eukaryotes have nuclear DNA on linear chromosomes. So when I say nuclear DNA, what I mean is their DNA is in a membrane-bound nucleus. So all of the DNA is contained in this specialized organelle that's found within the cell. And that DNA itself is also sort of organized on long linear chromosomes. So they're folded up into those sort of X shapes that you're used to seeing in textbooks. But if you were to take either end of that X shape and pull it out like you're pulling out a sweater, that string would pull out and it'd be a long linear piece of DNA that's not connected on either end. It's important to note, though, that in some of these organelles within eukaryotic organisms, there is circular DNA in some of the organelles. Specifically, mitochondria and chloroplasts have circular DNA. In addition, there are many multicellular eukaryotes. So typically when we think of eukaryotic organisms, we think of multicellular organisms, things like animals, big plants and trees, fungi, but it's also important to note that there are many unicellular eukaryotes as well. For the prokaryotes, typically the cells are much smaller, and typically they're entirely unicellular, okay? And when I say entirely unicellular, almost every single prokaryote we know of is unicellular, although there are a few, very, very, very few exceptions to that. For the most part, all prokaryotes are unicellular. If we were to blow this prokaryotic cell up and make it look much bigger, you'd see that there are no membrane-bound organelles or nucleus. So once again, the nucleus is the kernel, so to speak, that eukaryotes have that prokaryotes do not. This is before the kernel. There is no nucleus and no membrane-bound organelles. And the DNA in prokaryotes is typically organized on circular chromosomes that are connected from end to end. So typically those circular chromosomes are all folded up in wacky, weird ways, but if you were to pull it apart, it'd stretch out into one big hula hoop that's connected on either end in a circle. Okay, so let's look at some of the major traits that define bacteria, archaea, and eukarya. So first, I want you to answer this question. Of the things at the top, choices A, B, and C, which one is not a prokaryote? A, B, or C? If you said eukaryote, you're right. So the eukarya is its own group. Bacteria and archaea, however, are both prokaryotes. Okay, so let's go through each of these major groups and just play along with me, think on your own. For each of these groups, let's answer this first question. Does it have DNA in the nucleus? Does the bacteria have DNA in the nucleus? Yes or no? Does the archaea have DNA in the nucleus? Yes or no? Does the eukarya have DNA in the nucleus? Yes or no? If you said no for bacteria about DNA in a nucleus, you are correct. If you said no for archaea, you're also correct. They do not have a nucleus. A nucleus is an organelle. They don't have organelles. And eukarya does. What about organelles? Bacteria, yes or no? No, it does not. How about archaea? Yes or no? No, it does not. How about eukaryotes? Do they have organelles? Yes, they do. What about circular chromosomes? Bacteria? Yes, they do. So their chromosomes or their DNA is organized into a circular chromosome. What about archaea? Yes, they do. What about eukaryotes? No, they do not. Their nuclear DNA is linear. So the DNA that's contained in the nucleus, that's the major component or the largest percentage of the DNA in a eukaryotic organism, is linear. However, eukarya have mitochondria, and plant eukaryotes or photosynthetic eukaryotes that have chloroplasts will also have circular DNA within their cells. Typical cell size. So bacteria, large or small? They are smaller. Same for archaea. The eukaryotes are in fact larger or typically larger in terms of their cell size per an individual cell. How about types of reproduction? So your choices for this are asexual reproduction or sexual reproduction. What do you think for bacteria, archaea, and eukaryotes in terms of the types of reproduction they're capable of? Okay, so bacteria can only reproduce asexually. Same with archaea, they can only produce asexually. With eukaryotes, you can actually get a mix. Some reproduce asexually, most reproduce sexually. Some can actually reproduce in both ways. So it's a mixed bag with the eukaryotes. The important thing is that bacteria and archaea reproduce exclusively asexually. Eukarya can do both. Cell wall structure. So you can think of a cell surrounded by a cell membrane, and on the outside of that cell membrane is a cell wall that's sort of a protective structure that protects the individual cell. So as it turns out, there's specific structures for each of these different groups. Specifically, bacteria that have cell walls have peptidoglycan, which is a specific type of molecule that makes up the cell walls. Archaea can be varied, but they never have peptidoglycan, chitin, or cellulose making up their cell walls. And eukarya, if they have a cell wall, it will be either made of chitin or cellulose. So like the cell walls of a plant is made of cellulose. Animals, fungi can have a cell wall made of chitin, but they'll never have peptidoglycan making up their cell walls. And lastly, this thing called an RNA polymerase. So an RNA polymerase is just a very important biological molecule that's present in the cells of all these different organisms. They all have RNA polymerases. But the structure of that RNA polymerase was different for bacteria, archaea, and eukarya, such that bacteria have a smaller RNA polymerase that's only made of 5 subunits. So subunits are the units that make up this RNA polymerase molecule. So it's a smaller molecule for bacteria, whereas both archaea and eukarya have larger RNA polymerases that are greater than 10 subunits in size. Okay, so why did we go all through this? These are very important aspects that distinguish bacteria and archaea and eukarya from each other. And it's important for you to actually memorize each of these different aspects of these different groups of organisms. And so I'll expect you to know these different traits. And I'll expect you to be able to apply those traits to different scenarios. So, for example, let's pretend you just found a new organism. Its DNA is only in circular chromosomes. It reproduces asexually, and its RNA polymerase has greater than 10 subunits. Which of the following is best supported? If you said this organism is an archaea, you are correct. This organism is an archaea. So remember, archaea are the only ones that are going to have DNA just on circular chromosomes, reproduce asexually, but also have an RNA polymerase with greater than 10 subunits. So that's unique. Those 3 traits are unique, or having all of those 3 traits is unique to archaea. Let's try this one again. What if your organism has some DNA in circular chromosomes? It can reproduce asexually, and it has an RNA polymerase greater than 10 subunits. So if you said that this one is an archaea or a eukaryote, you are correct. So recall that eukaryotes have their nuclear DNA in a linear format, but they also have circular DNA in their mitochondria and chloroplasts. So a eukaryote can have both circular or linear DNA. It can reproduce asexually, so remember that both archaea and eukaryotes can reproduce or capable of reproducing asexually, and both also have an RNA polymerase greater than 10 subunits. So in this case, this organism could be an archaea or a eukaryote. You can't distinguish it at this point unless you went and actually looked at the nuclear DNA, whether it had a nucleus, and if it does have a nucleus and has linear DNA, then you'd be able to say, okay, this is a eukaryote. So let's look at a couple examples of archaea and bacteria. Specifically, let's discuss what they are, why we should care about them, when they evolved in the sort of deep geological history of planet Earth, and why they're so successful. So this is what we'll spend the next couple of lectures discussing, learning about some of the interesting biology in these groups, the bacteria and the archaea. So let's start off with what they are. So this phylogenetic tree on the right represents the two domains, bacteria and archaea. You can see the domain eukarya down here at the bottom in gray. And you can see that within these two major groups, we've got several different sort of kingdoms that represent the bacteria and the archaea. So I would expect you guys, or I will expect you guys to memorize the names of every single one of these groups and know some aspects of each of these groups. Ha ha ha. Just kidding. So really what I want you to pull from this is that there are several different kingdoms within each of these major domains. And the following. So which of the following is false regarding prokaryotes based on this figure? So this is hearkening back to that table we worked with. Which of the following is false? So E is false. They are not a monophyletic group. Remember, they are a paraphyletic group. So you got to snip the tree twice to get this group, the prokaryote, to fall off. It doesn't pass the one snip test. So looking at the structure of the typical prokaryote or prokaryotic cell, you'll see that they have a plasma membrane surrounded by a cell wall. So they'll have both a membrane and a cell wall. If you look at the DNA within a prokaryotic cell, it's big, so it's this dark blue sort of stringy spaghetti stuff that we're looking at right here, and it's folded into a bunch of loops, but it's contained in a circular chromosome. So if you pull this thing the whole way apart, it'll be like a big sort of hula hoop. And it's just floating around in the sort of liquid cytoplasm that fills up this cell. So there's no nuclear membrane surrounding that DNA. It's just sort of freely floating within the cell. You'll also find that many bacteria often have small circular additional pieces of DNA that are called plasmids or plasmid DNA. So this little sort of halo hula hoop of DNA that you're looking at right here, this is an example of a plasmid. And so this is different than the sort of circular chromosome that we're talking about here. It's a smaller piece of DNA. And they also have these little ribosomes that are floating around helping to make proteins. When we look at this sort of general form of a prokaryote, there's actually a ton of variation on this general structure. So we can find them in these sort of round things called cossi, and they can vary in size from 0.3 micrometers all the way up to one of the largest prokaryotic cells that we know about that's 100 micrometers, almost 200 or 300 micrometers in diameter. So just comparing the sizes of these two, they can vary in size a lot. They can also vary in shape, so they can be rods, or they can be filaments, or they can be spirochetes like we see right here. They can have flagella on the back. So there's a lot of variation on this general structure, but they're all, for the most part, unicellular and microscopic, very, very, very small. So why should we care about bacteria and archaea in general? Because they run this joint. They play extremely, extremely important roles in the functioning of life on Earth. They can affect human health. They have extreme habitat and metabolic diversity, so they can live in a lot of places that other organisms are not capable of living. They are masters of evolution, and I'll talk about what I mean by that in a few slides. And they're extremely abundant, the most abundant organisms on Earth. So let's talk about their effects on human health. So I think most people recognize that bacteria can have really negative effects on human health. A great example of this is the plague caused by the bacterium Yersinia pestis, or the Black Death is also what it's called. This is a bacterial disease that's spread through mice that infect fleas, and when fleas bite humans, they can spread that bacteria into humans. Humans end up getting pretty bad infections, often resulting in cough and infection of the lymph nodes. And towards the end stages of the infection and the disease, they start to go necrotic, where the skin and other cells in their body will die. And when they die or go necrotic, they turn black, like you see in the picture right here. And that's why it's called the Black Death, because it's essentially killing the tissue slowly. And in the past, before we developed good antibiotics and good vaccines against Yersinia pestis, against the plague, there were major outbreaks of this disease that significantly impacted human populations. So this is just population change in Europe over time, from 1,000 all the way up to 1,700. And what you see is in the sort of mid-1300s and mid-1600s, there were big outbreaks of the Black Death or plague outbreaks that really influenced the population. In fact, reduced the European population by almost 20 million people, or 25%, back in the mid-1400s. It's pretty significant. Obviously, some pretty significant effects on the population. I should point out that this x-axis or this y-axis doesn't go to zero, so it makes the effect look a lot bigger than it is, like you lost about 75% of the population, but really this is just 25% of the population. But it doesn't reduce or lessen the point that this has some serious negative effects on human populations. But we also have a lot of really, really great effects or positive contributions that bacteria and archaea have made to human health. For example, streptomyces are responsible for 2/3 of the natural antibiotics. So things like tetracycline, streptomycin, erythromycin, neomycin, cypermycin, chloramphenicol. These are all natural antibiotics that have been derived from bacteria. And in fact, on the whole, the vast, vast, vast majority of bacterial and archaeal species are either positive for humans or have no real effect on humans at all. It's a very rare exception of bacteria that's harmful to humans. And in fact, when we look at new studies about the human microbiome, we find that there are many really good and important species of bacteria that are necessary for human survival. So for example, if you look at the human gut, things like bifidobacteria, E. coli, and lactobacilli, these good bacteria that you see at the top here, these all play important roles in helping with immunity and processing nutrients in the human gut, and are really vital for human survival. And we're just starting to learn about all the positive effects that bacteria can have for humans. And so they're looking at other studies of the microbiomes of hair and oral cavities and nostrils and the esophagus, and we're finding that there's huge diversity in all of these different locations in terms of the types of bacteria that are there, and we're learning a lot more about how these things are both positively and negatively influencing human health. So bacteria are important for human health, and that's just a few of the ways that they're important. Let's talk about the habitat and metabolic diversity of bacteria and archaea and the prokaryotes. So it turns out that these things can live pretty much anywhere in terms of habitat diversity. We can find them in hot springs that are boiling hot, that really nothing else can survive in. Super saline lakes that are so salty but nothing other than prokaryotes can survive in. Freezing glaciers, super acidic mud pits, the human gut, and even radioactive waste where no other organisms can survive. Prokaryotes have devised interesting ways to survive in these different habitats, which gives rise to the question of how. How is it that they're able to survive in all of these really difficult and extreme environments? And the way that bacteria and archaea they can do that, part of that is through their extreme metabolic diversity. So for example, some bacteria can survive in hydrothermal vents where there's no light and there's no major nutrients or food supplies other than the sulfur that spews out of these vents. And they figured out how to use sulfur basically as a food source. Cyanobacteria are the original photosynthesizers. So long before plants evolved the ability to photosynthesize, cyanobacteria sort of solved that riddle and figured out how to do it. And they're the first organisms to be able to make food exclusively from light. So this is once again a very example of a diverse metabolism, being able to get food from different sources. And there's even some bacteria that can survive on oil. So they've released or tried releasing some of these bacteria into oil spills because they'll just eat that oil up. It's a great energy source for them. So once again, examples of this tremendous metabolic diversity, and all these different species of bacteria in archaea have specialized adaptations that allow them to get energy and nutrients from a ton of different sources. And this results in their abundance and ability to live in a lot of different habitats. So one of the ways that they're able to evolve all of these crazy different types of metabolism is because they are masters of evolution. And when I talk about masters of evolution, they're capable of actually exchanging DNA through asexual means. So they can do this through these processes called horizontal gene transfer, where individuals of the same and even different species can pass DNA off from one individual to another. So one example of this is a thing called conjugation, where you can have DNA passed from a donor to a recipient. And once again, these don't have to be members of even the same species. You can also have DNA transfer, this process called transformation, where essentially a bacterial or archaeal cell dies, it releases its DNA out into the environment, and then another bacterial cell that's sort of floating around will actually pick up that piece of DNA that's floating around from the environment. And so there's no need for this sort of messy and complicated process of sex that we see in eukaryotic organisms. It doesn't even have to be the same species. They can enhance their genetic diversity just by sort of picking up DNA from other individuals or the environment. So this is hugely advantageous in terms of the spread of alleles and traits and picking up new alleles and traits. Another really important aspect of bacteria and why in archaea and why we should care about them is their abundance, their overall abundance. So if you were to pick up an individual teaspoon of dirt, just to give you an idea, that individual teaspoon of dirt holds 1 billion microbial cells. So a huge abundance. Because the cells are so tiny, we can find tons of them in even the smallest little piece of dirt. And in fact, they compose half the biomass on Earth. So half the total sort of living cells on Earth by weight are prokaryotic. And if you were to line up all the bacteria and archaea on Earth from end to end, it would stretch longer than the Milky Way. So this is just goes to show you the huge amount of abundance that we have on these organisms. Because of that huge amount of abundance, they have a very important role in what's called biogeochemical cycles. So influencing the movement of nutrients and gases from the air to the water into the soil, and moving a lot of these important nutrients around, and play an important role in ecosystem level processes because they're so abundant. Yet we've only named 10,000 species, and so there's still a ton that we don't know about the archaea and the bacteria, about these prokaryotes. More about the abundance. If you were to look just in the human mouth, you would find over 700 species of prokaryotes. So they are insanely abundant in the sense that just in your mouth alone, you can find over 700 species. And in fact, 90% of the cells in your body are either bacteria or archaeal. So just take a minute to wrap your mind around that only 10% of the cells in your body are actually you. The other 90% of those cells are estimated to be bacteria. So you're more bacteria than you are you, which is crazy to think about. So this is just a sort of idea of what might be going on in your gut. It's a bit of a silly cartoon, but you get the idea here that there are tons of prokaryotes crawling all over you. And as I mentioned before, because of their abundance, they might have a lot of sort of play some important beneficial roles for the humans. And in fact, there's some evidence now suggesting that certain bacteria can help ward off obesity, where they find a different microbiome in people that are obese versus not obese. And it's gotten to the point now where people are starting to even create art. So this is just an example of an artwork where people are basically taking these sort of growth medium and having people touch their arms or different body parts and seeing what bacteria grow to create this interesting artwork. So that orange stuff that you're looking at, orange stuff that you're looking at here represent bacterial colonies that were isolated from a person's arm. So if we think about the prokaryotes, let's talk about this next sort of topic. When did they evolve? So we've learned what they are. We've talked a little bit about why we should care about them and their huge abundance. So when did they evolve? When did they appear on Earth? So I want you to spend maybe 2 minutes putting the following events in chronological order and how many years ago you think they happened. So go ahead and spend a little bit of time working on that. Try to put these in order. So now that you've completed that activity, let's look at this little map that represents the history of life on Earth according to the fossil record. So we can start here about 4.5 billion years ago with the origin of Earth and move forward to prokaryotes appearing on the screen. So Earth forms about 4.5 billion years ago. The first life we see form about 3.75 billion years ago. We see the first single-celled eukaryotes coming onto the scene a little over 2 billion years ago. We see multicellularity, so having multicellular organisms that are composed of many different cells interacting with one another, happening about 1.5 billion years ago. We have animals popping onto the scene about 500 million years ago. And lastly, we have humans popping onto the scene in sort of the last 200,000 years. So if we were to take this same geological time scale of these major sort of evolutionary events, and we were to squeeze them onto a 30-day calendar. So this is just to give you an idea of sort of like deep time and how much time has passed and how long have humans have been around. So if we put Earth forming on day 1 and then the 30th day on the calendar is present day. If we zoom in to the last day, we see Homo sapiens popping onto the scene in the very last 10 minutes. So just to put that in perspective, the entire history of humanity in terms of the 4.5 billion years that Earth has been around represents only 10 minutes if you're to squeeze all of that onto a 30-day calendar. So we've been around for a very small speck of time on, if you think about it on a geological time scale. Okay, so bacteria and archaea, remember the prokaryotes. When did the prokarya first appear on Earth according to this figure? If you said 3.5 billion years ago, you are correct. So this is prokaryotes popping onto the scene. The first prokaryotes evolved or appeared on Earth about 3.5 billion years ago. So they were among the first organisms, living organisms to appear on Earth. And it was about 1.7 billion years before eukaryotes evolved. So they've been around for a very, very long time. They were the first living organisms on Earth. So I'm going to stop there for now. I just want to point out upcoming readings and assignments that you should be keeping track of under Module 5 on Canvas. So these are all the readings that are posted. Make sure you take a look at those. And these are all the assignments that are posted with the due dates coming up. So take a look at those. I especially want to point out this pre-lecture quiz on photosynthesis, respiration, and redox reactions. Please take that pre-lecture quiz before you start working on the next lecture assignment. This is going to be important to review this stuff to help you with that. Okay.